Everyone, this is Ravindra Babu. Welcome back to my channel Silver 90s Bionit. In the last video we were talking about the types of RNA molecules where we discussed only the first one, that is tRNA molecule, but still two types of RNA molecules are pending. Those are rRNA and mRNA. In this video let us start with the rRNA molecule. Here rRNA is nothing but ribosomal RNA. As this RNA is a constituent part of ribosomes, hence the name ribosomal RNA. And this rRNA is a linear polyribonucleotide, but shows loops here and there like this. So this is RNA molecule, rRNA molecule, which is a polyribonucleotide, but here and there we can find loops like this. Loops will form wherever the complement nucleotide comes. Okay. And this RNA molecule is abundant RNA. Abundant RNA. Nothing but it is 80 to 90% of total RNA of a cell content, total RNA of a cell content. And this RNA molecule is considered to be as a structural molecule because this ribosomal RNA is a constituent part of ribosomes. Ribosomes, along with the rRNA molecules, they also consist of some proteins and rRNA molecules. Hence ribosomes are considered to be as nucleoproteins. Nucleoproteins means here these consist of nucleic acids, RNA and proteins, hence the name nucleoproteins. Viruses are also called to be as nucleoproteins, but these nucleoproteins and viruses are different. Viruses also consist a nucleic acid, either DNA or RNA, and a protein capsid. That is a different, and this is a different, but both are considered to be as nucleoproteins. Keep remember this. And here the ribosomes are of two types, we know very well. The first one is a 70S type of ribosomes, 70S type of ribosomes, to which we can find in prokaryotes. The characteristic feature of prokaryotes is having a 70S type of ribosomes. And the second type of ribosomes are 80S ribosomes. 80S ribosomes. Here, to which we can find in eukaryotes. This is the structure of a 70S ribosome, which consists of two subunits. One is a large subunit and the other one is a smaller subunit. Okay, so this is a large subunit of a 70S ribosome and this is a small subunit of 70S ribosomes. This large subunit is a 50S subunit and small subunit is just a 30S subunit. The 50S subunit consists of rRNA molecules such as 23S rRNA and 5S rRNA. And the small subunit consists 16S rRNA. This is a combination of different types of rRNA molecules that a 70S ribosome is having. And next, if we come to 80S type of ribosomes, here the 80S type of ribosomes also consists two ribosomal subunit. One is a large ribosomal subunit and another one is a small ribosomal subunit like this. The large ribosomal subunit is a 60S ribosomal subunit and small ribosomal subunit is 40S subunit. The 60S subunit consists of 28S rRNA and 5.8S rRNA molecule and 5S rRNA molecule. Okay, 3 types of rRNA molecules it consists, but here only 2 types of rRNA molecules. And a small subunit consists a 18S rRNA molecules. This is what the combination of different types of rRNA molecules in both 70S type of ribosomes and 80S type of ribosomes. These rRNA molecules of small subunits such as 16S rRNA and 18S rRNA are actually complementary to the Shane-Dalligano sequence of mRNA. The Shane-Dalligano sequence means, say this is mRNA molecule and this is the first triplet codon AUG and it is the 5' end, this is the 3' end. The portion of mRNA present before AUG towards the 5' end is called a Shane-Dalligano sequence. These rRNA molecules are complementary to this Shane-Dalligano sequence so that they can make the mRNA to bind with the ribosomes properly. Now let us move on to the last type of RNA molecule in the living system, that is mRNA. mRNA, here M stands for messenger. This is a messenger RNA because it involves the transfer of genetic information from DNA to proteins in the process of a central dogma of molecular biology. And this mRNA molecule is also a linear polyribonucleotides but without loops. The reason is the mRNA molecule consists of a 7-methyl guanosine, which is a unusual nucleoside present at a 5' end, so that any nuclease cannot break the mRNA. And at the 3' end, this mRNA consists of a polyadenylate tail. This is what we call poly-A tail at the 3' end. So that this poly-A tail, it restricts the mRNA molecule to become double stranded. It restricts the mRNA molecule to form a loops. Okay, this is what simply the structure of mRNA molecule, and on this mRNA molecules the nucleotides are in the form of triplet codons. This is a triplet codon, this is a triplet codon which specifies a particular amino acid, and it is a longer or larger RNA molecule than the remaining 2 types of RNA molecules, and the content of mRNA is just about 3% of total RNA content of a cell. The mRNA molecules are of 2 types. One is monocistronic mRNA, mRNA, which is present in eukaryotes. Where can you find this? Eukaryotes. This is the characteristic feature of one of the characteristic feature of eukaryotes. And second type of mRNA is polycistronic mRNA, to which we can find in prokaryotes. So here, in order to understand what is cistron and what are monocistronic and polycistronic, first you need to know the central dogma Central dogma of molecular biology. In central dogma of molecular biology, the genetic information it gets transferred from DNA to mRNA and mRNA to a protein or polypeptide chain. This is a DNA. During a transcription, here the genetic information it gets transcribed into a mRNA. And the genetic information present in mRNA it gets translated into a polypeptide chain. This polypeptide chain later by undergoing some conformational changes it may become as an enzyme. Enzyme involves in a particular biochemical reaction. That biochemical reaction is get expressed in the form of a character in the organism. This is what the central dogma of molecular biology. Here, what is a cistron? Cistron means a portion or a segment of a DNA molecule which is going to give a single polypeptide chain. A polypeptide chain is called cistron. Say this segment of DNA is a DNA cistron. This mRNA is also involving in the synthesis of a single polypeptide chain, so that this mRNA is considered to be as monocistronic. Okay, that is what about the cistron and monocistronic mRNA. This happens in the case of eukaryotes. And let us come to the central dogma of molecular biology in the case of prokaryotes. So here say this is a DNA molecule. This is a cistron of DNA, and which is by undergoing transcription giving a mRNA molecule. This mRNA by undergoing a translation it produces more than one polypeptide chain. Say this is it may produce 3 polypeptide chains which comes from a single mRNA, so that this mRNA is polycistronic mRNA. Here this happens in the case of prokaryotes. The function of this mRNA, what is the function of mRNA? Just by seeing this central dogma of molecular biology, you can say that mRNA is carrying the genetic information from DNA to polypeptide chains, hence the name messenger RNA. Thanks for watching this video. Please like, share and subscribe my channel.